Hello everyone. For those of you who don't know me, I'm Babak Faryabi, Assistant Professor of Pathology and a core member of Penn Epigenetics Institute. It's with a great pleasure to introduce Dr. Bing Ren, who is kindly joining us from San Diego today. Unfortunately, his in-person visit was kind of disrupted due to the new variant, but I'm sure you all equally will enjoy the virtual presentation. So I should confess that actually it was very challenging to summarize extraordinary contributions of Dr. Ren to the field of epigenetics, unless you would like to hear from me for the next 1 year. So I'll be very quick and only highlight a few snippets of his unparalleled contributions, and would let you to enjoy about the latest discoveries from his lab. Dr. Ben started his scientific journey in China before he moved to Boston, where he received his Master in Science and PhD in Computer Science and Biochemistry from Harvard. I believe this is very unique. He then moved a few blocks to Whitehead Institute for his postdoctoral training with Rick Young, where he invented chip-on-chip and used it to create one of the first genome-wide map of transcription factor binding. After his postdoctoral training, Dr. Ren has appointed to UCSD faculty in 2001, when he's currently the director of Center for Epigenomics, a member of Ludwig Institute for Cancer Research, and a professor of cellular and molecular medicine. Dr. Ren's research truly revolutionized many of our understanding about genome organization and regulation. His research group has continuously innovated transformative technologies and deployed them to answer questions that were we as a scientific community were not able to answer without those technologies. For instance, Dr. Ren has led the creation of first map of gene regulatory elements in human and mouse genomes as part of the ENCODE project. His team pioneered studies that unravel mechanism of genome folding, including discovery of topologically associating domain or TAT structures that you heard about in your courses. Dr. Ren's team also more recently innovated a number of technologies to investigate gene regulation at single cell resolution. And I think this is a topic that you will hear more today. Dr. Ren's transformative research has led to nearly 200 publications, which were cited astonishingly more than 75,000 times. That was very intriguing to see this morning. In recognition of his seminal contributions to the field of epigenetics, Dr. Ren's research has been recognized by several awards, including Chen Award for Distinguished Academic Achievement in Human Genetic and Genomic Research, and elected to American Association for the Advancement of Science Fellow. Without further ado, please join me to welcome Dr. Ren for today's seminar. I also would like to ask you to please mute your speakers so we have no interruption. Also type in your questions and we will discuss them as we move forward. Thank you. Thank you, Dr. Ren. It's all yours.
Thank you, Babak, for that very kind introduction. And I am very honored to give this seminar. I would have loved to give it in person and meet with the colleagues on, in this incredible campus. But as Babak said, it's probably not the best time to travel due to the pandemic. Despite so, I hope that this virtual seminar will still convey some of the key messages I want you to think about, and I welcome a discussion either during or after the presentation. So we're in the depths of the pandemic, COVID-19 pandemic. But let's not forget that the society also has a huge burden on numerous other diseases, most of which actually have a strong genetic components, such as cardiovascular disease, cancer, mental disorder, and the neurological disorders. This adds together incur millions of lives lost and tens of millions of life years lost in terms of productivity. And the question for us who works on life science is, what's the mechanism of this disease, and how can we find ways to treat, prevent, or cure such illness? Sequencing of the human genome really has been a landmark in the life science and allow us to identify genetics of landscape of complex traits and disease. And genome-wide association studies is a perfect example where sequencing technologies has advanced our understanding of the causes of numerous human traits and disease. And I think one of the key lessons emerged in the last 20 years was that non-coding DNA plays a major role in the initiation and progress of human disease, and also, of course, different traits. The most recent catalog of GWAS, encompassing thousands of studies, several thousand studies, identified hundreds of thousands of sequence variants associated with the traits and disease. Majority of them actually are non-protein coding. And that gave us a very, very challenging task. What do they function and how do they contribute to the disease? And this is exacerbated by two aspects. One is, what are the causal variants of each disease? When one performs GWAS studies, typically a flagging leading SNP is reported. But for each leading SNP, you have dozens of other SNPs that are in linkage disequilibrium, some of which are significantly associated. Others are just below the threshold. And they are probably equally likely to cause disease. So it's a major task to find the causal variants. And secondly, when you have a SNP that associated with the traits and disease, it's frequently unknown what cell types and what tissues are contributing to the disease symptoms or phenotypes. So these are the major tasks that that we confront every day. If we want to study mechanism disease, we need to have ways to go beyond the GWAS study. A paradigm that has emerged in the recent years is that non-coding variants function by disrupting transcriptional control elements and transcription of genes in disease-relevant cell types. Many examples have emerged, but there have been severe bottlenecks for us to advance this paradigm. Number one is we don't have high-resolution maps of regulatory elements that are working for most cell types. The target genes of annotated regulatory elements in the human genome still yet to be precisely defined, and transcription factors that are affected by variants at such regulatory sequences also are largely uncharacterized. So this bottlenecks really prevented us from going much further beyond having a list of non-coding variants associated with certain traits and disease. So what should we do about it? Today, I would like to describe three, I would say three steps, baby steps that we have made. Hopefully will make a contribution to help advance this field. I will first describe how single-cell epigenomics tools that others and we have used to allowed us to generate maps of regulatory elements that are cell type resolved and across the human body. And secondly, I will show you that a systematic study of transcription factor binding to non-coding variants allowed us actually to come up with better understanding of how transcription factors bind to DNA and distinguish between different variants. And thirdly, I'll go over very quickly some of the latest tools that allow us to go beyond non-coding variants, linking them to potential target genes. I would like to start by saying that epigenome is the key to address non-coding variants of the genome. Epigenome, as you're all quite familiar with, refers to the modification of DNA, a modification of histone proteins on in the nucleus and the chromatin fiber, and also refers to the chromatin structure, both in one-dimensional, such as open chromatin, and three-dimensional conformation, such as long-range chromatin contacts. So altogether, we refer to them as epigenome. And epigenome have now been extensively studied using sequencing-based tools, such as Illumina high-throughput sequencing, and tools such as ChIP-seq, ATAC-seq, DNA-seq now allow us to precisely map the location of such modifications to either DNA or protein, histone proteins. And that, in turn, generated maps of epigenomic modifications in the cell. The utility of such maps has really been shown in the latest ENCODE and epigenome roadmap project, in that they allow us to track the dynamic of chromatin state of DNA, especially at promoters and distal regulatory elements such as enhancers, and allow us to come up with a detailed annotation of regulatory elements in the genome. But previous epigenome assays only gave us bulk result, average of the bulk population, which is equivalent to this world map from the mid-ages, where it's there, but it's not precise. You have some of the continents, but you miss others. So it's incomplete, and it's also crude. What we really need is single-cell-based approach to examine epigenomes. And that's really in the recent last few years, we have seen rapid development in this space. Single-cell epigenomic assays allow us to examine the DNA or histone modifications at individual cell basis. And this is important because every cell shares the same DNA, but slightly different epigenomes. And study the epigenome in individual cells is really going to give us the necessary resolution for us to pinpoint the function of the epigenome in health and disease. There are roughly 3 major approaches that have been reported that allow us to do single-cell-based epigenome assays. The first one is plate-based. These are essentially miniaturized epigenome assays, such as high-C or methylation sequencing. Each cell is sorted into individual wells, and then you perform reaction in parallel manner. A second way is now droplet-based, popularized by the 10x genomics platforms. This allows you to look at beyond several hundred to thousands of cells to tens of thousands of cells in parallel. And for example, single-cell ATAC-seq, single-cell cut and tag have all been developed for this purpose. And more recently, a very exciting and new platform was described. It's called split and pooling barcoding or combinatorial indexing. This approach essentially generates DNA barcodes for each cell in a combinatorial fashion, where you combine, for example, multi-well reactions. You pull all the cells together, you redispense cells into multi-wells, and you perform this in multiple runs. In this fashion, you can basically generate millions of combinations of DNA barcodes, and that allow you to scale up the reaction to a very, very high scale. And this is also what we choose to develop many of the single cell genomic or epigenomic assays. We start by adopting a single cell combinatorial barcoding strategy invented by Jay Shanduri's lab from University of Washington. It's called Sky Attack. Basically, thanks to an effort of former postdoctoral fellow in my lab, Sebastian Prezel, we adopt this method, optimized it for frozen archival samples, and further introduced automation so that we can perform such assays in a very high throughput manner. We routinely profile tens of thousands of cells per sample with high reproducibility and low cost and very high signal to noise ratios. Using this strategy, we can generate an open chromatin at individual cell level. We also encountered significant challenges in data analysis. Unlike bulk ATAC-seq, single cell ATAC-seq data typically is sparse. You get several thousand to tens of thousands fragments per cell. Definitely not the whole picture of the open chromatin. So in order for us to process data from single cell ATAC-seq, a former student in my lab, Zhongxin Fang, developed a pipeline called SnapAttack. What this does is essentially the whole suite of tools added together from quality control, normalization, to clustering, and also peak finding. So this basically provides a complete package of a single cell ATAC-seq analysis. The major innovation here is the clustering of cells using a k-nearest neighbor graph. In this approach, we actually can very accurately identify cells based on the cell type that they group together. And once you have the cell groups, you can then aggregate cells from each cluster together. First, you can identify peaks shared open chromatin for each cell. And you can also annotate the cell type based upon marker genes, because typically cell lineage specific genes are showing open chromatin in the cell type that they are active, but not in other cell types. So using this strategy, we can now analyze ATAC-seq data. Another major improvement of SnapAttack is its high scalability compared to previous tools that we had. This new tool allows us to profile and process up to 1 million cells in parallel by optimizing the clustering approach. So this is the tool that we are now using for analyzing our single cell ATAC-seq data. The reason we want to go such high scale is because human body is complex, and we have to analyze millions of cells in order to have a comprehensive view of the different chromatin landscape in different cell types. So this is an ambitious project that just came out last week, but it took us more than 3 years to complete. This is a project that initiated by a graduate student of mine, Jake Hawker, together with a postdoctoral fellow, Kai Zhang, and with the help of Sebastian Prezel and Alan Wang, both of which are at Center for Epigenomics. We began with human tissue, primary tissues that were collected as part of the GTAX and the ENCODE collaboration. So the donors are selected to be of normal, and their cause of death is not related to any significant disease. And the tissues were collected fresh very, very quickly after death and is stored in appropriate condition. So we were fortunate to gain access to these tissues and perform single cell ATAC-seq. Altogether, over 30 different tissue, primary tissue types from up to 4 donors. We collected single cell ATAC-seq data from over 600,000 nuclei. Using SnapAttack and the iterative clustering, we grouped them first into 30 major cell groups corresponding to some of the known major cell types. And after this, we also, for each of the major cell groups, perform a second round of cell clustering to further uncover the subtypes or subclasses of cells. For example, from gastrointestinal epithelial cells, we could uncover cell types such as colon epithelial and small intestine, enterocytes, goblet cells, and other cell types that some are very abundant, but others are quite rare in the tissue. So this gives you a general view that the single-cell TAC-seq strategy that we employ really uncover not only the cell types, but also reflect the proper cell composition of each organ or tissue. As I mentioned, that by aggregating cells from each of the cell clusters, we can infer the cell types for that cluster. For example, this is a marker gene corresponding to the small intestine goblet cells. And you can see that this marker gene is accessible at the promoter and in the gene body, but not accessible in any other cell lineages. This is another cell marker where it's accessible in enterochromaffin, but not in any other cell types. So using such marker genes, we were able to annotate each of the 111 cell types that we uncovered. And they group into generally this tree structure based upon their similarities. This is a hierarchical tree built by comparing the open chromatin landscape of each of the cell class. And you can generally distinguish epithelial cells from endothelial cells and mesenchymal cells. And even from the, this is the mesendoderm cells, this is the fibroblast lineages. We can identify fibroblasts that are unique or biased towards certain organs. And this is epithelial cells. Again, you can see many different subtypes of epithelial cells that have very strong preference in different organ and body types. One of the key motivation for us to do this is we want to identify cell type specific regulatory elements. So we identify peaks of each of the 111 cell types and add them together. We come to roughly 900,000 open chromatin peaks coming from 111 different cell types. Compared to a catalog of candidate regulatory element previous annotated by the ENCODE published last year, we added nearly 400,000 new element to this catalog and recover close to 60% of that catalog. The new element that we added, they actually are likely real because 95% of these new elements were previously associated with active chromatin state in one or more tissue types, and they show generally conserved sequence compared to random. Using unpublished ENCODE data, we found 13% of them to be tested positive for enhancer activities in a single lung cancer cell line. So this indicating that these new open chromatin elements are likely function as enhancers or other regulatory elements. We also integrated our adult tissue atlas with a fetal human tissue atlas previously published by Jay Shanduri's lab. This from 15 fetal tissue types combined with 30 adult tissue types out together. We have accumulated 1.3 million total profiles. We processed them all together in the same pipeline and identified a total of 222 cell types, half of which are adult specific, half of which are fetal specific. And you can basically also identify peaks for each from each of the cell types and group them based upon the cell type specificity. So this is 222 cell types and 1.15 million total elements put together in this heat map. And you can basically see the relationship between each of the cell types are shown in this hierarchical graph. And this, the color represent the different lineages such as lymphoid, myeloid, endothelial lineage, and other type of lineages. And bottom here, this is the heat map of each element. The color represent the normalized chromatin accessibility. So by and large, most of the elements that we identified here in this study show cell type specific open chromatin signatures. And basically these signatures, when you look into them, they reflect the type of genes that are expressed in that cell lineage. Compare this again to the ENCODE catalog published last year. We show that we added altogether nearly 600,000 new elements to this catalog and uncover close to 70% of the previous catalogs. So now, using this map, can we address some of the questions that I posed at the beginning? First, can we use this catalog to infer disease-relevant cell types of the common traits and disease? And secondly, can we use this catalog to identify likely causal variants involved in human disease? I like to address them together. So the major hypothesis is that the regulatory elements in the disease-relevant cell types should be enriched for the likely risk variants associated with disease and traits. So in this graph, I highlight some of the major, this is the approach that we are taking. As I mentioned in the beginning, each GWAS result actually identify the lead SNPs, and the other true causal SNP is probably buried nearby or buried beneath this threshold. So the key question is, which one is the causal variance and what cell types could be involved and contributing to the disease trait? The framework of thinking was essentially proposed by Alkes Price and Benjamin Neal from Harvard 5 years ago. So in this landmark study, they devised a method called LD score regression analysis. This involves the following 4 steps. First, you partition all the SNPs, including the lead SNP and the SNPs in linkage disequilibrium, into functional categories based upon, say, epithelial cell regulatory enhancers or muscle cell enhancers. And you assume that the LD to a category is enriched for the variability is coming from this enrichment. So you can use a chi-square test to test whether the SNPs in linkage disequilibrium with the lead SNPs are enriched in any of these functional categories. And this is achieved using this regression approach called LD score regression analysis. And once you identify such enrichment, that will inform you that the particular functional categories, such as a cell type specific landscape, would likely be the contributor to that disease. And using this strategy, we then examined 240 complex traits. So each of the traits and disease is selected because they have more than 20,000 cases. And we asked whether there is a strong association or enrichment of the risk variants in any of the enhancer or chromatin landscape from the 222 adult or fetal human cell types. The heat map here is indication of FDR, basically this is a coefficient, this regression coefficient. If it's strong, highlighted in red color, that basically means FDR less than 0.01%. And you can see that most of the traits that I listed here have a strong association with one or a few cell types, except for a subset that are associated with multiple cell types. So I'll give you one example, allergy asthma. This is a trait shown here. It's strongly associated with the T cell. And because we have more than 200 different cell types, this type of data give us very precise prediction that the T cell gene regulation is likely an important factor contributing to the genetics of this disease. Another one, coronary artery disease, again shows very strong enrichment in or association with a smooth muscle, two types from the vascular smooth muscle in adult, and also cardiac parasites. These are not well understood, but now we have some candidate now to go after for the molecular or genetic mechanism of this very widespread traits. Type 2 diabetes, this is known to be have a strong genetic component, previously linked to beta cells. We also, through this single cell ALICE, we strongly link type 2 diabetes to multiple cell types in the pancreatic islet, not only beta 1, beta 2 cells, but also alpha cells. And this is, I believe, is the adipocytes. COVID-19, this is something that we always wanted to know, why some people, when they were affected by COVID-19, they largely have presented mild syndromes or no symptoms, but then a small number of us could end up in the hospital and ER room and even on ventilator. So this Jiva study published last year, have identified a number of loci associated with COVID-19 hospitalization. And our study now shows that the genetic of those severe cases of COVID-19 is strongly associated with alveolar type 1 and alveolar type 2 cells in the human lung. So these are the two types of cells that are in the inner lining of the alveolus. And the alveolar type 2 cells is the one that express the famous receptor protein called ACE2, and that allows the virus spike protein to interact and to enter the body. So our genetic association appear to suggest that this is a cell type that should be looked very closely in terms of having severe symptoms versus mild symptoms. I want to discuss atrial fibrillation a little bit more in depth. This is a trait that affect quite a large number of population, and it's a major risk factor for heart disease. The symptom is that people with this traits tend to have a heart beating arrhythmia. You can see that a normal heartbeat is regular, maybe 1 per second, but then atrial fibrillation have this irregular heart beating with fast and slow pace intermixed. And this actually could cause severe heart disease because of the lack of synchronization between different heart chambers in pumping blood. We showed, as I earlier, so this trait is strongly associated with cardiomyocyte here. This is basically the p-value log 10. So cardiomyocyte is likely the culprit of this disease. Not very surprisingly, but now we can actually look at the risk allele generated from the fine mapping and ask which gene and which regulatory elements harbor these risk alleles. One of the enhancers or candidate enhancer identified in cardiomyocyte happened to have 2 such likely causal SNPs on top of it. And we therefore went look further, and we showed that this candidate enhancer indeed is capable of driving reporter in vitro differentiated cardiomyocyte. And the risk allele replacing the alternative allele reduced that reporter activity by more than half. This is all in the sense in agreement that this 2 risk allele, as I shown here, is likely the causal allele for this trait. We further knock out the risk allele, actually the enhancer harboring the risk allele in embryonic stem cells by CRISPR-mediated genome editing, and differentiate these cells into cardiomyocyte. So wild type cells express normal levels of this protein and the gene, and knockout of the enhancer, unsurprisingly, reduced the expression of this gene by more than 70%. What is this gene? It's a gene called KCNH2. It codes for a potassium channel protein. What potassium channel protein does is allow potassium ions to pump out of the cell, because that basically during the action potential, you first have a large influx of sodium ions, and that basically elevate the membrane potential from negative to positive. And then you open up a potassium channel to allow potassium ions to go out of the cell membrane to balance out and restore the negative membrane potential for the cell. So in cardiomyocytes with wild type enhancer, this action potential lasts roughly 300 milliseconds, but in the cardiomyocyte with enhancer knockout, this action potential lasts more than 900 milliseconds, almost a second long. So this is, I think this tells us that this risk allele is causing the misregulation of the potassium channel protein, reducing its expression, and therefore likely responsible for the irregular heart beating phenotype that we see. So I hope I have convinced you that this single cell atlas of chromatin accessibility really provide a valuable resource for us to look into the genetics of a whole battery of human traits and disease. And now we have the tool to study which cell type and which genes are likely affected by the non-coding variants. That is a significant step moving forward than just having a list of non-coding SNPs that we don't know what they do. I'd like to use the remaining time to address two other bottlenecks I highlighted in the beginning of the talk. Another bottleneck that we want to study, we want to overcome, is predicting which transcription factors are likely affected by the SNP. We typically consider this as very following. Basically, if you have two alleles, risk allele and non-risk allele, maybe they act by blocking transcription factor binding or by encouraging transcription factor binding. So it's very important to learn which transcription factors is affected by it. In the bioinformatics field, we are very much used to this representation called position-weighted matrix for transcription factor binding specificity. What this icon or symbol means is that if you have a high A or G here, that means transcription factor prefer recognizing this basis at this location. If you have very low bar, that means this they don't care. And this is a quantitative matrix, and you can compute the score, which is a sum of how deviate a sequence from this idealized matrix. And this score basically gives you a prediction. If you have a delta, means if the alternative sequence is lower than the reference allele, then you predict that this transcription factor is binding less well to this sequence. This is how we typically represent transcription factor binding. The problem is we never tested this in large scale, how accurate this can predict transcription factor binding in vivo, especially to non-coding variants. So to address this problem, I collaborated with Yossi Tapali, who is now at Cambridge University. The project was initiated and carried out by a very motivated and talented postdoctoral fellow, Jay Yan, and collaborated with a bioinformatics computational graduate student, Yin-Jiang Cho, and Andrea Santos, who was a visiting graduate student at the time. So what we did was we carry out a high throughput transcription factor DNA binding assay by using a pool of DNA oligos incubating with recombinant transcription factors in a 96-well plate. So basically each well we carry out individual transcription factor binding to the pool of DNA oligos. Then you perform elution and wash. And you sequence the bound DNA after each round of binding, elution, and wash. You do this multiple rounds. And you can imagine that as you go through more and more such wash elution, binding, and sequencing, the true binding substrate will eventually become enriched in the pool of DNA that you come out from this solution. And if you have two alleles, one is bound by, preferentially bound by the factor, another is not, you will see this diverse trend with the bonding substrate to be gradually gaining in abundance, while the unbound, dispreferred allele to be increasingly underrepresented in the final pool of DNA oligo. We actually can perform a thought experiment and derive a relationship between this score. This is area between the two curves. And we call it preferential binding score. And there is a linear relationship between this score and the Ka differences of dissociation constant of the transcription factors between these two alleles. So this is convenient. So now we have used this approach to interrogate it, 300 or so transcription factors across 100,000 SNPs in the human genome. The SNPs were selected largely from T2D associated locus, mainly because this is funded by a type 2 diabetes project, but we also included a large number of other SNPs that are just in the candidate regulatory element near this GWAS loci. So altogether, we have roughly 95,000 SNPs that we interrogated using this very high throughput manner. And afterwards, we calculate this preferential binding score for each pair of alleles and identify those that are showing preferential binding by one or more transcription factors. End of the day, we identify a total of 11,000 such SNPs that show a preferential binding to one or more transcription factors. So that's the result. It came out earlier this year. I just want to summarize, basically using this result, we can now test systematically how well the PWMs are predicting binding of transcription factor to different alleles. And this is a precision recall curve. You basically want to see this curve, if it's like ELK3, that means it has a very high sensitivity and specificity, using our empirically determined result as the gold standard. So some factors are really predictive. Their PWMs are really predictive. But then there are other factors where the PWMs are extremely poor in making such predictions, such as FOXA and IRF3. This may relate to the fact that these are binding very short sequences and the motifs were not defined that well. So this is a summary. Out of 129 transcription factors where we can perform such assessment, we found that only 24 reach a threshold that we consider satisfactory, while a vast majority are not so predictive. And you may ask, why? I mean, why should we believe your result than the PWM? This is what actually got me convinced that our results are valuable. So we basically ask from all the preferentially bound SNPs, do they show a enrichment for the most likely causal SNPs? So this is basically to show that for type 2 diabetes, we have about 100 GWAS loci, but hundreds or thousands of SNPs in linkage disequilibrium. But by using a Bayesian inference, we can actually identify so-called likely risk alleles using, as for example, here, this is posterior probability of association. This is threshold basically, if it's above 0.1, it's become increasingly likely that you're finding the causal SNPs. If it's approaching 0.9, you're very possible to find the real causal SNPs. So we actually see a very strong enrichment as we use this threshold higher and higher of the empirically determined, the preferentially bound SNPs in our assay, in experimental assay. But if you use PWMs for the same factors, you don't see at all this enrichment for type 2 diabetes associated variants. this suggests that actually our experimentally determined assays are actually very informative. Encouraged by this, we went one step further. So Yun Jiang, who basically a computational biologist, he basically used a gapped K-mer SVM. It's a machine learning approach pioneered by Mike Beer, by Leslie Christine from Memorial Sloan Kettering many years ago. What they basically, this framework does is to basically allow you to come up with a model that better predict how transcription factor can differentiate between the two alleles. So we were able to use this transcription factor binding assay result from previously published data sets and trained with our model to come up with a highly accurate so-called delta SVM approach. So this allows us to then to make distinguishings between the two alleles at much higher accuracy than the PWMs. So this is the result. Basically from, before I said from 122 transcription factors where we have enough data to make assessment, only 24 out of 189 exceed this PR of the threshold of 0.75. But using our newly derived delta SVM model, we actually can make such accurate predictions for 94 of them. So this is a significant advance compared to the PWM approach. So now I think this approach should be adopted, in my view, to systematically looking at all the transcription factors against all or a large set of transcription non-coding SNPs to allow us to better predict how transcription factors distinguish between non-coding SNPs. And then finally, I think this is the question that bothers a lot of us, which is once you identify the risk SNPs or risk variants, what genes do they affect? And there is there are many ways to do this. And I think one of them, a very exciting development in recent years is this activity by contact model proposed by Jesse Ingres, who is now assistant professor at Stanford University. What this does is basically consider histone modification at the distal elements, as well as 3D chromatin contacts. So basically, simply by using the product of this histone modification and chromatin contacts, he was able to make fairly good prediction of target genes of transcription factors. And that's, sorry, of enhancers. So basically on this, the major problem though is we need histone modification data at single cell level and also chromatin contacts at single cell level. Can we achieve that? Earlier this year, my lab published a paper where we described one such method called paired tag, which was invented by a very talented postdoctoral fellow, Chen Shui Zhu. What this assay allows us is to perform profiling of histone modification together with gene expression from the same cell at high throughput from millions of cells in parallel. Here is the rough scheme of it. In the first step, we combine the single cell platform with the so-called cut and tag platform that Steve Hanikoff lab developed. In this assay, you incubate cells with antibody against histone modification, and then add in protein A, TN5 fusion protein, where TN5 could introduce double-strand breaks at the site where this antibody is binding to, which is the histone modification mark. In the same cell, we also perform reverse transcription using poly A to convert messenger RNA into cDNA. Once we carried out this assay in bulk, we can then perform combinatorial indexing using a strategy called split and pull. In this approach, we sequentially add multiple combinations of DNA barcodes in 96-well plates so that we can basically add 96-well 96 different barcodes to both the cDNA and the open chromatin cut by the protein ATM5, and then mix everything together and then perform the same reaction again after dispensing into a new 96-well plates. So we can do this in multiple runs and creating millions of different combinations. Afterwards, we can split the RNA encoding library and the DNA encoding library and then perform sequencing and identify the levels of transcription along with histone modification marks. And this is basically a result from an experiment that we Chen-Shi performed on mouse brain. You can see that the complexity of the library is quite high. We can basically reach the same level of library complexity as 10x. And the histone modification marks, different marks depends on the antibody, give us different complexity. Lysine 4 monomethylation, lysine 9 trimethylation, we can reach tens of thousands of unique reads per cell. But for others like K27, it's more mediocre, medium. But the key point is that we can now separate cells based on RNA reads. And for example, the mouse brain, now using our strategy, we can first separate them into 22 different cell types corresponding to excitatory neurons, inhibitory neurons, as well as glial cells. And for each of the cell, we can then aggregate the histone marks and come up and identify the different histone modification landscape for that particular cell. For example, lysine 4 monomethylation, trimethylation, lysine 27 acylation from the same basically cell types are plotted here. This is really giving us a chance to now assess the relationship between histone marks at distal element and gene expression at the locus. And we can basically see a positive correlation between lysine 27 acylation and gene expression, and negative correlation between 27 trimethylation and gene expression across cell types for many of the genes. So the genes that show very strong, significant positive or negative correlation between these marks and histone and gene expression are highlighted here in blue and brown curve shadows. And they frequently correspond to the same gene. And that's because, like oligo 1, like oligo 2, they're regulated by this essentially same enhancer that when they are active, they are associated with lysine 27 acylation. But when they are inactive in other different in other cell types, they are the same enhancer is occupied by lysine 27 trimethylation. So this is essentially a negative and positive mechanism of regulating these two lineage-specific transcription regulators. So we actually can uncover such relationship between distal element and the target genes in mass. This slide just to show that the pair tag data actually give you equivalent single-cell RNA-seq data. This is basically 10x droplet sequence RNA-seq performed on the same type of cells, tissues, and you basically uncover exactly the same type of cells. And this basically this comparison shows that the Pearson correlation coefficient of genes are highly similar, reading out from both platforms. It also shows a comparison of pair tag with single cell cut and tag. Essentially, single cell cut and tag essentially is cut and tag, but using 10x droplet-based sequencing. And we show that this pair tag actually has 10 times better coverage per cell basis, and is more accurate in revealing cell type classification, and give you 100 times higher throughput, 1 million cells per assay versus tens of thousands cells per assay. And it's truly multimodal instead of just single modal. So given the time, I will just very quickly go through this final technique that we developed. This is basically to profile chromatin conformation from the same cell. But how do you achieve that? We achieve that using a combination of bisulfite sequencing and 3C mapping. We call it methyl high C, and also my collaborator, Joe Acker, independently developed the same method called methyl 3C. Essentially, you identify chromatin contacts from the same cells, but the key problem is how do you interpret this data? The data is sparse and very difficult to identify loops and domains because of that sparsity on individual cell basis. My collaborator Ming Hu and his postdoctoral fellow Amang, working with Miao Yu, a former postdoctoral fellow in my lab, developed this method called SnapHiC, which I will not go through detail because it's published. But this method allows us to map chromatin loops between enhancer and promoters or between different anchors from as few as 100 or 200 cells. The same data, if you use conventional methods such as hiccups, only give you a few dozens of loops. But with our method, you can actually uncover tens of thousands of loops. And the accuracy measured using bulk high-C data is actually quite satisfactory. So we apply this method to single cell high-C data set generated in Joe Acker's lab from mouse, this is human brain. And we basically, from roughly a dozen cell types, we were able to call loops and the number of thousands to tens of thousands. This is the number of cells per cluster, and this is the number of loops in the units of, this is 5,000 to 10,000. And this kind of data allow us to really identify in physiological context distal elements and target gene connections. For example, this is a site from the ApoE locus. We know ApoE is one of the highest risk factors for late onset Alzheimer's disease. This risk variance shown here is associated with ApoE gene expression, but it's very specific to astrocytes and not in other cell types. So this gave you the fact that this gave you an idea that astrocyte may be the culprit of where the ApoE misexpression is contributing to disease. So this is the final slide. I'd like to summarize what I've said. We have built a single cell analysis of chromatin accessibility for the human genome across hundreds of cell types and millions of elements. And that allow us to really make inference on the disease relevant cell types and identify likely causal variants for many, many human traits and disease. We also now develop a new scheme to identify transcription factor binding on SNPs that perform better than conventional PWMs, and develop new tools of single cell multiomics to help link non-coding risk variants to their target genes. And I'd like to close by thanking my colleagues. I mentioned their names, I spoke, and I want to specifically mention the funders that listed here for contributing to the work I just described. Thank you. I know I run out of time. If we have 10 more minutes, I'm happy to answer questions.
Thank you, Bing. I'm sure people already have a couple of questions here. So if people have time, we will continue with the questions for the next few minutes.
Sure.
I think the first question is from Klaus. Is more of a, I don't know what is the question or is more common. Basically, Klaus is stating that one of the reasons that the in vitro selects might not always represent the in vivo PWM from ChIP-seq is that the in vivo, there is chromatin accessibility and cooperative binding. From ChIP-seq, we know that there are many theoretically good sites that are not actually bound by FOXA2. So would you comment on that?
Yeah, that's a very good observation. I completely agree that I think many of the PWMs derived from ChIP-seq data may actually be a combination of multiple factors that contributing to the lack of predictive power for in vitro data. But another factor that I didn't have time to go over is the fact that the PWM assume independence of bases in their contribution to transcription factor binding. But we know that this is not always true, and the support vector machine framework essentially does not require that two bases nearby are independent. In fact, it explicitly captured that, and that might actually be one of the reasons contributing to that model being more accurate in making predictions.
Thank you. So the next question is from Joe Zhao. He is obviously saying amazing talk. So the question is, in addition to assessing the TF binding for those GWAS SNPs, I wonder if you have examined the impact of SNPs to chromatin accessibility using your single cell ATAG data, particularly if any SNPs may have dominant effects on chromatin accessibility.
That's a very good question. We haven't explicitly done this, but in our most recently published this paper in Cell last 2 weeks ago, we did incorporate this CLAX SNP data into the prediction model, basically predicting which transcription factor may be perturbed by the SNP. We did not test the prediction, but that's something that I think we should do next. Essentially ask whether those are affecting the chromatin accessibility. We now have more data to directly test it. Thank you for making that point.
So the next question is from Sora Yoon. She's asking, how did you set the gold standard of loops to estimate the accuracy? Is that from simulation data or?
That's a very good point. We simply use the bulk Hi-C data and call the loops using hiccups. These are like billions of read pairs. So we assume that is the gold standard. And then we basically downsample the essentially single cell Hi-C data from to 100 cells, 200 cells, 300 cells. And we use this bulk Hi-C gold standard data to ask how accurate and how sensitive our loop callers are recovering that bulk data. So that's the gold standard. So we described that in this Nature Method paper more closely. So happy to go over more details of it.
Thank you. So the next question is, have you compared your SELEX results to an in vivo assays, such as MPRA, to assaying allelic effects on TF bindings / enhancer activity?
Yes, the answer is yes. We described that in the paper. We showed that SELEX results predict much better the MPRA result from different alleles than PWMs.
The next question is from our own Jennifer. She's asking, thanks for showing these cool signal cell data. In your pair tag analysis of enhancer target gene predictions, have you looked to see how many of the predicted pairs are also looping in ensemble Hi-C data? It might tell us once and for all to what degree loops might be required to help link distal common variants to target genes.
That's a great question. I'm not sure whether it can tell us once and for all, but we did do such comparison, and roughly 50%, 60% of the predicted pairs are supported by Hi-C data from bulk tissue. Anyway, so it's actually quite encouraging. However, I would say it won't probably solve this problem once and for all.
So the next question is from Garrett. Is a follow-up to Klaus's question. He's asking, TFs often bind to elements that are lower affinity than their optimal binding sites, something that has also been referred to as enhancer sub-optimization. Do you think this affects the interpretation of the results?
Yeah, I'm aware of those beautiful work from Mike Levine and others. I would say our result doesn't really directly address the issue. I think what we did was an in vitro assay, and transcription factor binding parameters as estimated from our model, what we try to see is whether PWM accurately recapitulate this in vitro assay result. And I think our conclusion is that the SVM machine learning model actually better capture that than PWM. In vivo, how does this model affect our interpretation of the enhancer usage and the effect by SNPs? That's not what we try to address, but I think that's probably the next step. With this type of large-scale data sets, we can potentially better address that in a physiologically and in a human population.
So maybe before going to the next step, I kind of ask my own question. So having the large compendium of single cell ATAC you generated, have you ever assessed how many cells we need to measure in order to identify rare cells? And whether the enhancers are a better marker of the cell type or the gene?
It's a very good question. Our data suggests that you need probably tens of thousands of cells or even more from any given heterogeneous organ and tissue to identify cells with abundance of 1 to 2%. If you want to, for example, in the colon tissue, we tried very hard to identify the stem cell compartment and we failed. And I later learned that those stem cell compartment is only about 0.1% in the tissue. So we most likely need to scale up even further 10 times to be able to capture those stem cells in the colon. So yes, it's an issue that we have to address. The good news is the single cell ATAC-seq is quite cheap. We only seek, I think my estimate is sequencing cost is only on a few cents per cell. So if you want to, we can really scale up to millions of cells to do in-depth analysis.
Thank you. So the next question is from Pankaj. He's asking, do you have a list database of all your novel GWAS SNP to target gene maps? Are these captured as part of EBI GWAS catalog?
So yeah, in this paper that we published recently, we have a supplementary table where we link roughly 3,000 such GWAS SNPs to targets. So we have such predictions. But the caveat is this is based upon crude data and about the high-C data. So it's not necessarily accurate. So I want to make this precaution right now so that you don't blame me when you find the wrong gene.
So the next question is from Hal. He's asking, have you benchmarked ABC model prediction using Sci-High-C results?
Not yet. Good question. This is something that we are very keen on doing. There just haven't been a lot of single cell high-C data yet.
Correct. Okay, so probably the last question before the speaker gets to the next meeting. Since methylations and enhancers and promoters can impact TF binding, would you combine single cell methylation and single cell ATAC data? Be helpful in validating cell-specific PWMs associated with TF binding and activity?
Yes, absolutely. I think that's a very good point. And indeed, I'm collaborating very closely with Joe Ecker. I mean, as you know, his lab is an expert and probably where it's best placed to do single cell methylation assays. So we're very closely working together to map human brains using the combined single cell ATAC and the methylation studies.
Okay, so thank you so much, Dr. Ren, for such an amazing talk. And yeah, that's clearly shows from all the questions. And I hope you enjoy the rest of your visit.
Thank you very much. Thank you all for your attention and interest in our work.
Thank you. Have a nice day. Bye-bye.
Bye.